Biochemical test for bacterial identification. Importance of the accurate bacterial identification. Determine clinical significance of particular pathogen. Guide physician care of patient. Determine detection of antimicrobial resistance. Determine appropriate type of antimicrobial therapy. Determine whether susceptibility profile are unusual. Determine if organisms is a risk to other patient, hospital, public lab work. Differentiate on basis of nutritional requirement, metabolic capabilities. Differentiate on basis of nutritional requirement, metabolic capabilities. Bacillus test to determine effect of bacillus on an organism. Differentiate the beta hemolytic streptococci, streak, suspect pure culture colonies onto BA plate using a aseptic technique. Place a bacillus disc on the streak area. After incubation, measure the zone of inhibition. Streptococcus pyogenes, Streptococcus agalactiae. Biochemical test. Test to differentiate group D streptococci and other streps. Group D strept enterococci grow in 40% bile presence and hydrolyze esculin in the presence of bile or bile salt. Group D strep will hydrolyze the esculin by product of hydrolysis. React with indicator ferric ammonium citrate. Expect result E is positive. Bile susceptibility test. Test differentiate the Streptococcus pneumoniae from other alpha hemolytic strep. Bile or desicolate rapidly lyse pneumococcal colonies. Lyse is dependent on presence of intracellular autolytic enzyme. Bile salt lower surface tension between cell membrane and medium, accelerating natural autolytic process. Positive colony. Disintegrate negative intact colony. Negative. The CAMB test. Detection of diffusible extracellular protein. Christie, Atkin, much better soon produced by group B streptococci. Enhance hemolysis of the sheep erythrocyte by Staph aureus. Positive Streptococcus agalactiae. Negative Streptococcus pyogenes. Differentiate between both of them. Carbohydrate fermentation test. Phenol red broth test. Test determine ability of the organism to ferment various simple carbohydrates, sugars, glucose, sucrose, or fructose. Determination characteristics are used in identification of bacteria, particularly the gram-negative enteric gut bacteria. Fermentation, a metabolic process in which final electron acceptor is an organic molecule. Each medium has a single fermentable carbohydrate added to the peptone medium. A small tube, Derham tube, is inverted and placed in each large test tube of liquid medium. The inverted tube traps any gas product. Phenol red is added as pH indicator. Phenol red turn yellow below pH 6.8 and darker pinkish red above pH 7.4. Carbohydrate fermentation. Phenol red broth test. Interpreting the result. Yellow acid production produce a color change from the red to yellow, indicating that the organism is capable of metabolizing the sugar in the tube. Acid gas, yellow plus gas bubble. Fermentation of the sugar is indicated by a color change to yellow. Gas is trapped in the Derham's tube, replacing the medium in the tube. A bubble indicates gas production. Negative fermentation can be indicated two ways. No color change in the tube means that the sugar was not utilized by the organism. 2. Color change to dark pinkish red. This darker color indicates an alkaline or basic metabolic product, which is due to the utilization of the peptone rather than the sugar. If the tube is read within 48 hours, the darker red color would be an indication of negative fermentation, although result is usually recorded as alkaline. Carbohydrate fermentation. Phenol red broth test. control Staph aureus acid, Xenomys vulgaris acid gas, Xenomys aeruginosa negative, E. coli negative. Catalase test determine catalase enzyme production. Catalase in catalyze hydrogen peroxide to water and oxygen. Key identification test for many gram positive, catalase positive micrococci, staphylococcus, catalase negative streptococci, streptococcus and enterococcus. Place a drop of 3% H2O2 onto a clean slide. Mix a small amount of unknown colony with into H2O2. Effervescence bubble, O2 gas. Bacteria produce catalase. Failure to produce effervescence, no bubble, no catalase present. The Staphylococcus aureus Enterococcus faecium test may be performed directly on agar slant. Citrate utilization test determine ability of an organism to use citrate as sole carbon source, Simon citrate agar sodium citrate as a sole carbon source, and ammonium ion as the sole nitrogen source. Bromothiamol blue turn from green at natural pH 6.9 to blue at pH higher than 7.6 basic or alkaline. Citrate utilization test determine the ability of an organism to use citrate as a sole carbon source, Simon citrate agar sodium citrate as sole carbon source, and ammonium ion as a sole nitrogen source. Bromothiamol blue pH turns from green at nature pH 6.9 to blue at pH higher than 7.6. If the citrate is utilized, the growth will produce alkaline product, changing the color of medium from green to blue. Coagulase test detect ability of certain Staphylococcus species to clot plasma. Coagulase production is characteristic of Staph aureus. Useful test for identifying these gram-positive catalase positive cocci. Place loophole of test organism into tube of plasma. Second control tube incubated with sterile saline or left uninoculated. Clot appear within 30 minutes if the coagulase present, Staphylococcus epidermidis and Staphylococcus aureus. Clot appear within 30 minutes if coagulase present. The test is read at interval up to 24 hours. ELK test demonstration of toxin production by Cyanobacterium diphtheria. A filter paper strip impregnated with diphtheria antitoxin is poured beneath surface of Columbia agar before it's hardened. Strain to be tested non-positive, negative toxogenic strain are streaked on the surface across the plate at right angle to the antitoxin. After incubation, plate examined for presence of fine precipitating line at 45 angle to the streak. Presence of precipitating strain produce toxin react with the antitoxin. This is the toxogenic Cyanobacterium diphtheria, non-toxogenic Cyanobacterium diphtheria. Methyl red test. Test to identify bacteria that produce stable acid end product by means of mixed acid fermentation of glucose. Combination media used MR-VR broth induced peptone, glucose, and phosphate buffer. Bacteria that are able to perform a mixed acid glucose fermentation produce large amount of stable acid. The pH indicator methyl red is added to at 48-hour culture. If the pH less than 4.4, indicator turn red positive, a yellow color. pH greater than 6 is negative, and orange color indicating a pH between the two usually require further incubation. Oxidase test. Test the presence of bacterial cytochrome oxidase respiratory enzyme. Cytochrome oxidase catalyzes transport of electron from donor compound to final electron acceptor. Useful in distinguish the oxidase negative Enterobacteriaceae from Pseudomonas aeromonas. Rub sample of bacterial colony onto a filter paper impregnated with tetramethyl phenyl diamine dihydrochloride. Examine the slide for result. Purple color endophenol enzyme present no color change is negative test. This test phenyl diamine artificial electron donor redox dye in its reduced from is used to reduce cytochrome oxidase. Optocene test to determine effect of optocene erythrodroborane hydrochloride on an organism. Differentiate pneumococci from alpha hemolytic streptococci. Streak suspect pure culture colony onto half BA. Streptococcus pneumoniae or Streptococcus viridans using aseptic technique placed on optocene disc on the streaked area after incubation. Measure zone of inhibition. Positive zone of 14 mm or greater 6 mm disc. Negative no zone of inhibition. Equivocal any zone less than 40 mm. Salt tolerance test determines the ability of the bacteria to grow on high salt concentration. This is used to differentiate enterococci positive from non-enterococci negative. Test the heart-brain infusion, both containing 6.5% sodium chloride, also small amount of glucose. Visible turbidity, negative no turbidity. Sulfur reduction in dole production motility. Sulfur reduction test to identify bacteria capable of producing sulfur. This is particularly important in determining certain genetic enteric organisms. Hydrogen sulfide can be formed by putrefaction or anaerobic respiration. Medium containing cysteine, an amino acid containing sulfur and sodium trisulfate plus peptonized iron or ferrous sulfate H2S react with iron or ferrous sulfate forming a black precipitate. Black precipitate present positive H2S production, no precipitation negative test. SIM test sulfur reduction in dole production motility. Indole production to identify bacteria capable of producing indole using tryptophanase. It's one component of IMVIC test for differentiating Enterobacteriaceae. Tryptophanase can convert tryptophan to indole, ammonia, and pyruvic acid. Indole is the metabolite identified by this test when Kovac reagent which contain hydrochloric acid and diethyl aminobenzyl aldehyde and amyl alcohol. A red layer will from when indole is present. Positive red layer and negative no color layer. Sulfur reduction in indole production motility. This medium can be stable, inoculated with an inoculating needle to indicating motility. Alwar agar concentration in the medium allow limited movement of the motile bacteria from the area of the step. Motility will be detectable as diffuse growth radiating from a stable line. Motile positive from H2S production. Triple sugar iron test to differentiate bacteria based on their ability to ferment to glucose, lactose, or sucrose to reduce sulfur to hydrogen sulfide. Primarily used to distinguish morphologically similar bacteria or Enterobacteriaceae, all of which ferment glucose to an acid end product. TSI agar containing peptone, glucose, sucrose, lactose, thiosulfate. pH indicator in phenol red, yellow. At a pH less than 6.8, red above 6.8. It's prepared on an agar plant with a deep pot to provide for anaerobic growth. It's inoculated by stab in the agar but followed by the fishtail streak of the slant. Urease test. Differentiate organisms producing urease. This test is particularly useful in the distinguishing the genus Proteus from other enteric bacteria. Urea test media, either broth or slant containing urea, and the pH indicator phenol red. This indicator turns to the dark pink magenta color when the pH is greater than 8.4. When urea is hydrolyzed, ammonia will be produced, raising the pH of the medium above 8.4. A positive reaction is dark pink, and a negative reaction will be either yellow or orange. Uninoculated control Proteus vulgaris E. coli. Vegas Prescour test to identify organisms' ability to produce acetylene from a degradation of glucose during 2 to 3 butadiol fermentation. Useful in differentiation between members of the Enterobacteriaceae. Vegas Prescour test use the same MRVR broth. Some fermentative organisms don't produce enough stable acid to lower the pH of the medium. For these organisms, the chief end product of the glucose metabolism are acetone and the 2,3-butadiol. After 48 hours of incubation, barite reagent A, alpha naphthal, and barite reagent B, potassium hydroxide, are added to the sample. After gently shaking the tube for aeration, formation of a red color will indicate a positive reaction. No color change or a copper color are negative result. Rapid biochemical identification. There are around more than so different biochemical tests. Fermentation tube, many different compounds as carbon source added to the number of possible differential tests that can be done. All these tests take time, use a lot of tubes and the material, and the result of many tests can be difficult to organize and make sense. Clinical micro lab must be able to identify sample quickly. The microbiologist must do many tests in order to identify an organism accurately. Manufactured test kits commonly used to rapidly enterotubes, Bioscience, many other manufacture. After incubation, a few drops of reagent are added to some of the compartment to complete test. Each color change and observation is recorded on a sheet provided with an enterotube. Each outcome is given a point value and the points are added up. The final number is used to look up the name of the isolate in a master handbook provided by the company. In hospital, more sophisticated rapid ID systems are used. Often process is automated, including inoculation. Automated system, more tests can be done. Multiple samples can be run simultaneously, and the result can even be read by optical sensor and analyzer by the computer software. Although this testing seems like make identification simple, there are limitations. Many organisms don't grow well in these kinds of tests. Tests rely on good growth during the incubation period. Example, Mycobacterium, a very slow growing organism, have a fastidious growth requirement. A rapid ID test has not been devised for this organism. More traditional methods are still used. Stacks and rope also more difficult to identify.